Yeah, it's coming. You can start the program.
Good afternoon, everyone. We welcome you all to our seminar series. So today is National Science Day, which is celebrated to mark the discovery of the Raman effect. C.V. Raman became the first Indian and the first non-white person to win the Nobel Prize in Science for his illustrious discovery. So today we celebrate the spirit of scientific inquiry that led to the discovery, not just of Raman effect, but also any other discovery in the field of science. Today's event is also a part of Azadi Ka Amrit Mahotsav, which is the initiative of the Government of India to celebrate and commemorate 75 years of Indian independence, our glorious history and our achievements. Now, without further delay, I'll request Dr. Kartiki Desai to kindly introduce our speaker for today.
Thank you, Dr. Alpana. Welcome everyone and good evening to all of you, teachers, students, and also our colleagues. It is my great pleasure, happy National Science Day, first of all. It is my great pleasure to introduce my colleague, Dr. Sandeep Singh. Dr. Sandeep Singh has done his PhD from the National Center for Cell Science in Pune. He was a postdoctoral fellow at Moffitt Cancer Center in Tampa, Florida, and he also joined NIBMG just a few months after I did, which was way back in 2012. I think since when he has been working on the current topics that he's going to discuss today. He received the intermediate fellowship and a research grant from Wellcome Trust DBT India Alliance. His interest, of course, is to understand stem-like cancer cells, particularly in reference to oral carcinogenesis and oral tumors. So without much delay, I would like to hand it over to Dr. Sandeep Singh, and he's going to talk to us today about intratumoral heterogeneity, the non-genetic causes and consequences. Dr. Sandeep, over to you.
Thank you, organizers, Dr. Kartiki Desai, Dr. Alpana, and of course our director, Professor Andhra Mitra, for giving me this opportunity to talk on this auspicious day of National Science Day. So my best wishes to all the listeners today about for the National Science Day. And thanks, Dr. Kartiki, for giving such a nice introduction also. So my topic today, as was said, that intratumoral heterogeneity, non-genetic causes and consequences. So as I would just like to introduce the field in which I am working on. This is in the field of cancer, and my interest specifically is to understand heterogeneity in the for a specific cancer which is very prevalent in India, the oral cancer. So if you might have noticed, the oral cancer which occurs in Indian people is because of tobacco chewing. They for the tobacco chewing, they keep the tobacco between the lip and the gum, and they develop cancer in this area. This is called gingivabuccal oral cancer. NIBMG is heavily involved in understanding, cataloging the genetic mutations which is occurring in this gingivabuccal region of oral cancer. Through this ICGC International Consortium for Genomic, International Cancer Genome Consortium, for this, which is involved in cataloging all different cancers which is occurring in people in the world. So the gingivabuccal oral cancer is a very aggressive disease. People die within 5 years with very high rate. And the aggressiveness of gingivabuccal oral cancer is as high as, like, if the patient is diagnosed with just the local lymph node metastasis, their chance of survival drops by 30%. So this is an aggressive cancer. We have understood the cancer in a much deeper way, knowing that the type of mutations they must be harboring. And also these studies have highlighted how heterogeneous the patients can be with respect to the mutation as well as gene expression patterns. So when I started to work in this area, the research goal which I kept for myself is to delineate and target the cellular and molecular basis of tumor heterogeneity. And just a second, if I can, yeah. And the three different arms which I am focused on is the functional heterogeneity in genetically identical cells, functional heterogeneity due to the genetic alteration in cancer cells, and the functional heterogeneity which can be achieved by the tumor because of the microenvironment, and we call it as biology of solid tumors. So I would just like to introduce you about the area of cancer heterogeneity. When the field started, we were mostly focused on understanding how a person's same tumor is different from one to another. So if these patients are treated in the clinic, not every patients are basically responding similarly to the given treatment. So basically that suggested that the tumor can be different from one another between the patients. So with that idea, this cancer genomics field started to catalog all those mutations which are most prevalent among responders to the therapy versus the non-responder to the therapy. As the field moved ahead, it was understood that within individual tumor also there are multiple different kind of disease. So that became the basis for defining what kind of clone within the tumor will be having the property of drug resistance, or which clone can be responsible for giving a metastatic disease or the relapse in the cancer of the cancer. So now the field has moved further. Now we are trying to understand within the genetically identical clones of within a person's tumor, there are different kinds of cells are there which is varying from each other based on their gene expression patterns or the protein the abundance of the protein which these cells might be making. So this is what I am calling it as non-genetic heterogeneity, because this is mostly occurring within a genetically identical clone present in patient's body within the same tumor. And so as we have understood cancer in a much bigger way now, we understand that the cells within the tumor or cells within our body as such is dependent on each other. Just like in a balanced ecosystem, the predator and prey, the predator is dependent on the prey to survive, and there is a unidirectional flow of energy in the balanced ecosystem. If this is the case, and that is prevalent in cancer also, it may be understood that if you remove one of these components, which may be the most crucial component to make to keep the balance of the ecosystem, we can perturb this ecosystem, and that will be devastating for that particular ecology. So if the tumor is an existing ecosystem itself where a lot of different kind of cells are there, which may be balancing each other, which may be helping each other to survive and to remain there with the changing environment or with the aggressive nature of the cancer. So if we identify a particular cell type, which is the most crucial cell type which is maintaining the whole ecosystem within the tumor, then probably that will help us targeting the cancer in a much better manner. So with this idea, the cancer stem cell model comes, where just like a normal stem cells, which is responsible for maintaining the homeostasis of the tissue, where the stem cells can self-renew and differentiate into different cell types and maintains the balance of that tissue or repair the tissue whenever it is damaged. With this similar analogy, cancer stem cells are also there, which is basically mutated normal stem cells. The normal stem cells accumulate mutation, and because of the accumulation of the mutation, they become cancerous. And if they are cancerous, we can call it as stem-like cancer cells. And these cells self-renew each other and also differentiate to form the bulk cells, which is forming the tumor. So if we think that this is happening in a balanced ecosystem within the tumor, so if we identify a cell type which is responsible for maintaining or responsible for making this ecosystem, which will be a stem-like cancer cells, because these are the cells which can self-renew each other. It has a long-term regeneration ability, and it has the property to generate all different kind of cells which is present in the bulk of the tumor cells. And therefore, this is suggested to be the most important cell type, which should be targeted if we want to win our war against cancer. And with this idea, the whole cancer stem cell field has started to move to identify, to have a proper identification of cancer stem cells for targeting them in an aggressive cancer. So I'm just giving you some pioneering example, which has suggested that how important the cancer stem cells can be to generate heterogeneity in tumor. So here, this is an example which is taken from a paper in 2011, which was in Nature Biotechnology. So here you can find one cell which is there, which is GFP labeled. So we can actually see that this single cell can generate all different kind of cells which may be present in the tumor. So if you take these cells, inject into the mouse, you will be able to generate tumor which is heterogeneous. How do you know it is heterogeneous? Because this is the tumor which is generated, and at least for two different markers, keratin 20 and Ki67, the authors have shown that the two different populations are being generated, even though when the tumor is generated just from the single cell. So if it is generated from a single cell, it is clonal, it is having a same genetic makeup, but the tumor is heterogeneous. So the marker which is used here is EpCAM positive, which is the marker for the epithelial cells, and the cells are CD44 positive. And when it is CD44 negative, the heterogeneity is the CD44 positive has generated the tumor, and it has generated CD44 negative population. So if you take the CD44 negative population, you inject into the mouse, it does not have the tumorigenic ability. So the cancer stem cell model suggests that the cancer stem cells, the cell which has this long-term regenerating ability because of the self-renewal and differentiation, are the only cell type which is responsible for generating tumor, and the bulk of the tumor cell, which is tumor non-tumor, the differentiated population, they are basically non-tumorigenic. However, in the same year, the group in the MIT, the Piyush Gupta et al., basically it is Bob Weinberg's group. In 2011, they have published this paper in Cell, which is telling us that the cell which is unique, they are clonal, having the CD44 marker. What could be the reason that it could generate CD44 negative cells? So if you start from a mixed population, you separate these population into different cell types. For example, this is the breast cancer. So here the breast cancer is divided. Breast cancer cells can be separated based on the specific cell surface marker into stem-like basal cells and luminal cells. The stem-like basal and luminal, the stem-like is the stem-like, basal and luminal is the differentiated population. However, after some time, you start to see these populations again coming in the system. So basically, they are suggesting that there is the generation of heterogeneity is stochastic. It is not regulated just because of the noise in gene expression, and you start to see all the other population in the system when you start from the 100% pure population. So this is what was suggested as the mechanism of generation of non-genetic heterogeneity in tumor. That is because of the stochastic mechanism, stochastic nature, behavior of the cell, the heterogeneity can be achieved over time. Okay, So with this background knowledge, I had to start with gingivabuccal oral cancer. So when we started to look which cell we have to work on, we did not find any cell line which can be made available to us to start working on the gingivabuccal oral cancer. And so we took the challenge. We established our own. We established the cell line by our own in the lab, by collecting tumor samples from nearby hospital, Tata Medical Center, with the help of our collaborator, Dr. Rajiv Saran and Dr. P. Arun. And these cell lines are now available in the lab. The nature of this cell line is published recently last year. So, both these cells has shown a very distinct behavior and distinct nature at the genetic makeup. So with this cell line, I wanted to start to understand whether the heterogeneity is there and whether the cancer stem cell model can be studied. So we relied on three different phenotypic marker. One is CD24, CD44. These are cell surface marker, and ALDH1, which is aldehyde dehydrogenase enzyme, which is the intrinsic property of the normal our tissue stem cells for basically detoxifying our cells by converting the aldehydes to carboxylic acid. So aldehyde is toxic; it becomes carboxylic acid, and that can be used by the cells. So naturally, cancer stem cells has very high expression of ALDH1 to keep themselves free from these kind of toxic material. So by combining these three markers, the cancer stem cells have been defined for oral cancer, and these are some examples. This is from Professor Sushant Roy Chaudhary's lab in 2016. They have also used CD44 high, CD24 low cells. It's the cell which has high expression of CD44 on the surface, whereas a low expression of CD24 on the surface. They basically show the behavior of the cancer stem cells. Similarly, among the CD44 positive cells, there is a small proportion of ALDH high cells, which is also shown by Max Vicha group, which is also very prime group in the field of cancer stem cells with respect to breast cancer stem cells. They have shown that ALDH high population in CD44 positive cells, they may represent as cancer stem cells for oral cancer. So for us to start with, we had this CD44 positive, CD24 negative, ALDH high as our definition for cancer stem cells. So it is also very popular in very different kind of solid tumors, and that was the basis for us to actually just go ahead with these markers to identify oral cancer stem cells. So I'm just giving it a very in brief. How do we identify aldehyde dehydrogenase activity in the cells? This is based on the flow cytometry experiment, because we ultimately wanted to perform flow cytometry. So this ALDEFLUOR assay, which is a commercial kit available from a company, which is depending on using the substrate, which is this one, body P aminoacetaldehyde. This is basically is used as a substrate by the cells. If they have higher expression of aldehyde dehydrogenase, this substrate is converted into this body P amino acid. This is body P amino acetate. So this amino acetate of body P is more fluorescent than acetaldehyde, and that is the principle for identifying these population. So if there is more fluorescence, we can identify because we are using flow cytometry. And this is the inhibitor of aldehyde dehydrogenase activity, which is DEAB, diethyl aminobenzaldehyde. And if the cell has DAB, even though they have high aldehyde dehydrogenase activity, the activity will be inhibited. So we can see a clean plot in flow cytometer. However, if the inhibitor is not added, then the cells are able to convert this to BAA, and then we see more fluorescence, and that is how we can gauge this population to say that this is the population which may be having high aldehyde dehydrogenase activity. So by combining these three markers, CD24, CD44, and aldehyde dehydrogenase, we could see a very specific phenomenon among different oral cancer cells. So we combined our cells, which is established from gingivabuccal oral cancer, and also we got the cells from University of Pittsburgh, which is basically established from floor of mouth. So both these cells, both these kind of cells are used for this assay, where we are finding that the 24 negative. So as you go higher on this scale, the CD24 expression is going up. As you come right on this scale, the CD44 expression is going up. So you can see that all the population which you seeing here is all CD44 positive. However, we can segregate CD24 low and CD24 high population. So and then these cells are also performed for ALDEFLUOR assay, so we can actually see if the cells are becoming aldehyde dehydrogenase, if they are having higher aldehyde dehydrogenase activity. So as anticipated, we had seen that CD24 low population, which is actually reported as oral cancer stem cells, they have high aldehyde dehydrogenase activity. So it was relieving to us that yes, we are also seeing the high aldehyde dehydrogenase activity as per the literature. However, it was exciting to us because CD24 high population in each of these cell lines, you can see that they are also having aldehyde dehydrogenase activity. So the result of all the populations are given here. For our understanding in the subsequent slides, I have put these colors to code these cells for the phenotype. So whenever I say red, this is aldehyde dehydrogenase activity high cells, which is a CD24 negative. So this is what is actually available in literature, that CD24 negative ALDH high is basically the oral cancer stem cells. When it is orange, it is ALDH low, the cells which are here, the ALDH low population, and 24 negative. So the population which is coming out from these gates. And the LDH high 24 positive, if the cells are 24 positive and it also has the LDH high activity, they are green. And if it is ALDH low of 24 positive, then it is blue. So you can see that even the 24 positive, the 035, this is the line which we have established in the lab. This is solely CD24 positive. There is no 24 negative population, but still it has the cancer stemness and it has this green population in it. So that was not only the case for the established cell line. We collected the patient samples, and also the patient sample showed that 24 positive ALDH high cells are present even in the tumor with varying frequency from patient to patient. So the question was, the CD24 high ALDH high, is this also a stem cells? And what is the relationship of this 24 high ALDH high with the other cells? And what is the function of this? And how does it relate to the clinic? So with these questions, we started to work. And whenever you see any culture, any plate which we have under the microscope, these four populations always coexisted with this pattern of frequency. The blue is maintained at the highest frequency, red and green is mediocre and the lower frequency, and orange is always at the middle. So when we see that this kind of frequency distribution among the population, the first thing which will come in our mind is maybe they are growing faster or slower. The cell which is at low frequency, maybe they are slow growing. The cell which is at higher frequency, they might be fast growing. For that, you can do dilution assay. You take the cells, you stain them all equal, and then you let them grow. And if as they grow, the dilution, the dye which is stained gets dilute, and then you can do flow cytometry to see how many cycles of division it might have gone to dilute the dye. So the red, orange, green, and blue, all four populations, they were stained, sorted and stained with this dye. However, we did not see any significant difference in their proliferation ability, suggesting that they are all equally proliferating. Also, they were equally forming the colonies in the plate. So these are the two most common assays which is done in the labs to perform or to show that the cell has long-term proliferative ability And if they have the long-term proliferative ability, they are the stem cells. So we did not see, and it was disheartening for the student also to see that the hypothesis that the red population should have been giving the colonies, should have been the slow-growing population, that did not occur. So we went ahead and started to look for their repopulation ability. If the cell has the stem cell property, then it will be having the property to generate all different kind of phenotypes which you see. And if it is more committed or differentiated, then they will lose the property. So this is the 4 phenotypes which we are talking about. Now the left axis is the ALDH activity, the 24 is on the right axis, and the population can be separated. And by flow cytometer, you can see that the population is separated at very high purity. The red, green, blue, orange, all these populations are separated with a very high purity from each other. And then they were allowed to grow for 10 days after sorting. We plated them back as a pure population and then allowed to grow them for 10 days. In this 10 days, there was a different ability for these populations to generate the heterogeneity. The red population, which is supposed to be a stem cell population, which is used as a stem cell population for breast cancer, oral cancer, colon cancer, this population was able to regenerate this heterogeneity which we are talking about. The green population, which is also ALDH high type, which was of our question that whether it has the stem cell property. It did not generate the red population, but it mainly generated the 24 high type, means 24 ALDH high and ALDH low population. The ALDH low population 24 positive, it mostly remained themselves 95%, and the orange population, which is the 24 negative ALDH low type, is mainly produce only ALDH low type, which is orange and blue population. So 24 negative cells became 24 positive, and so on. So that suggested that the red cell has the most, the most potent to generate the population, whereas the blue is more specific. Means they have got the specification. They have achieved to the specification step of differentiation, whereas the orange and the green population, they are kind of committed progenitor cell. This is what the suggestion which came out from this experiment. So to understand whether they have the stem cell property or not, we did the sphere formation assay. The sphere formation assay is a very specific assay where if the cell has the long-term self-renewal ability and they can differentiate, they can form, they can survive in the culture for long, then they will be able to form a sphere. So these are adherence independent condition, and they are all, the only cells will survive and thrive here which are serum independent. So as the stem cell property is the serum independent property, the adherence independent property, and they because they self-renew and regenerate, therefore they can form the colonies in the non-adherent condition. So when the cells were separated, the red, orange, green, and blue, they were able to form the sphere. All 4 were able to form the sphere. And not only for the first generation, these spheres were collected, dissociated again, and then plated back, and they could form the secondary sphere also. So they have real long-term regeneration ability, just like the stem cells. So all 4 population, even though it showed more specification and these more commitment, but they still remained, they still showed the self-renewal stem cell-like property. We challenge them to form the sphere at as low as 5 cells, and it can be seen that the red, which is the most potent and published cancer stem cell marker, it was forming the maximum frequency of the sphere formation, was maximum efficient, and followed by orange, green, and the blue was the least. But what can be visible here is that all 4 population achieved 100% sphere forming efficiency when they were as low as 50 cells per well. So this suggests that the stemness may not be only present in the red population, but may also be distributed in all the other phenotype. How it is distributed, we estimated from the experiments. Out of 3 cells, there is 1 sphere forming cells for the red. Out of 6, there is 1 sphere forming cells for orange. Out of 11, there is 1 for the green, and out of 23, 1 for the blue population. And the similar results were observed in the other cell line which is tested. So the sphere forming, the cancer stem cell frequency is basically highest in the red and the lowest in the blue, but still the emphasis came that we should not ignore it just because it does not form the sphere at the higher efficiency. We took the challenge and further did the repopulation assay from these spheres. We collected these spheres and grew them at single sphere per well. So you can see that this is the microscope where you can see the sphere, and this is the student Kavya. She is collecting these spheres from the individual sphere from these wells and then plating them individually in the wells to form these colonies. Once the colonies formed, this basically remained as we had observed for the 10 days of repopulation assay. So the red population generated all the 4 population, the orange, mainly orange and blue, the green made the green and blue, and the blue remained mostly blue. So this is how the difference is. You can find that there is huge similarity even when the spheres are generated for 10 days and then the colonies were generated for next 20 days. The specification, commitment, and potency did not change for these population, and because of that, we could actually derive this model where we are saying that the red population, which is a potent cancer stem cell population in many other solid tumors, so CD24 low ALDH high population, is able to generate all heterogeneous population. However, when you take green, it mainly forms green and then it also forms blue. If you take orange, it mainly forms orange and it forms blue. If you take blue, you see that these arrows are not reverting. It is mainly forming blue. There is a slight plasticity we are seeing, means the blue is forming the green and the orange is forming the red, which was in the non-treated undisturbed growth if you have for the culture. We took help from our friend Mohit Jolly from Indian Institute of Science, Bangalore, to suggest us the trajectory by the mathematical formulas. He used the trajectory Markovian model of transactions of the population, which was used originally by the Piyush Gupta's paper, which I showed you in the previous slide. So with the same model, he could generate the trajectories of these population, where if you start from the red 100%, eventually you by time the orange, green, and blue start to come up. The red goes down, the blue is the maximum population. If you start from the 100% blue population, it remains blue with a slight visibility of the other population. If you start from the orange, you see the orange comes down, the blue becomes more. And if you start from the green pure population, the green comes down and the blue comes up. So this is basically a mathematical presentation of the result which I just showed you in the previous slide. And if you take these population and you challenge them to repopulate or to grow in cisplatin. Cisplatin we use is 2 micromolar. It is not killing these cells, so the cells can survive. So it is basically non-lethal dose of cisplatin, and it is just inducing whatever the effect the cisplatin has to give to the cell. So if you start from the red population, you basically see this phenotype. But if you start with the red population along with cisplatin, the stem cell population remains high. Similarly, if you start with the green population along with cisplatin, you see that ALDH high population, the green population is remaining higher. The more interesting is that when you have this orange population or the blue population, which is ALDH low type, which is more specific, it is forming its own type only. But in cisplatin treatment, it has started to form higher percentage of the CD24 positive ALDH high population. So all these cases, the ALDH high population is significantly more if the cells are repopulating in presence of cisplatin. So this is again mathematical representation of the same data, where if you start with the untreated red population, you get all these. But if you are treating the cells in cisplatin, so these arrows, the orange turning to blue goes away, the red turning to green goes away, the blue turning to green is basically upregulated, and the orange turning to red is basically upregulated. So the cells have completely overtaken this repopulation ability of the cell, and they want to keep this red and green population at much higher levels. Okay, so and if you have the mixed population, you start to see that the frequency of the green population is much higher, and we know that reason is that the molecular reason, which I have not shown here. These cells express very high level of drug-resistant pumps. So this is all suggesting that the CD24 positive population is crucial population for oral cancer. This is one report which is in 2016. It suggests that CD24 expression is a predictive biomarker for cisplatin resistance. And this is this is much more recent, where the CD24 positive is identified as a novel stem cells in the palpodentinal mucosa, which is responsible for teeth production in human and basically mouse. So that suggests that CD24 positive is also a stem cell population. So to understand that yes or no, they have the stem cell property, we again desorted cells and performed RNA sequencing on a population specific manner. So here in this list we have a huge list of stem cell regulators, and you can see that the red, orange, green, and blue, they have very specific pattern of gene expression. The red population is regulated for this set of genes. The red and orange is for this set of genes. All kind of cells are expressing this kind of set of cells. So the gene expression pattern, although the cells are originated from the same, they are just dividing and under the transitioning to a different phenotype. But while the transitioning, they are changing their gene expression pattern. So these are different gene expression states of the cell. The gene set enrichment analysis suggested that the green population, the red is generated from the green is generated from the red population. Once it is 24 positive, the 24 positive cells are more differentiated. They have started to express cells which is involved in keratinization or quantification. So they are undergoing differentiation, but still. So what is the if the cells are undergoing differentiation, how they are maintaining stemness? While we were scratching our head for to answer that question, this paper came in 2019 from the cell stem cells in the cell stem cells from Offerud Klein's lab, where they have basically single cell RNA sequencing is done for the basal layer of in the buccal mucosa. The basal layer is the niche, the home for the stem cells which forms this buccal epithelium. So they have sequenced these populations and found that the cells are not only stem-like cells are there in the basal layer of the buccal mucosa, but also differentiating populations are also there. So basically, the basal layer is mixed of the stem cells and the hybrid state of stem cells, which has initiated the process of differentiation, but still maintains the stemness. So this became the clue for us, where we are seeing that the red population is generating green. The green is showing the differentiation pattern, but maybe the green cells have still maintain the stemness, because this is the natural process of maintaining the hybrid state of stemness in buccal mucosal stem cell niche. So we took this, our signature, the red, the genes which are upregulated in red or the gene which are upregulated in the green population, and mapped our gene expression pattern with the gene expression which is suggested for these population, the stem population, the differentiated population. So we found that the the genes which are upregulated in red population, they are matching more with the stemness, stem cell signature of this buccal mucosa, and less with the differentiated cells. Whereas the green upregulated cells, they started to match with the differentiated population, but also it had good match with the stem cell population. So it clearly suggested that this is our the green, the blue, and the orange. You see that they both have almost equal matching with the stem and differentiated population, suggesting that these populations are although differentiating but still maintaining the stemness in them as a hybrid cell state, as it is shown by this paper. We performed this gene expression analysis for the embryonic, the adult tissue stem cells, the embryonic stem cells, and our population is showing the match. We did perform RT-PCR to show this phenomenon. So you can see that the red population has highest level of SOX2, whereas the green and blue start to express SOX9. And so on. Then the cytokeratin markers, which are basically the basal layer marker, they are not lost in any of these populations, the red, green, or green and blue. So all four populations, they have the basal layer, the cytokeratin marker, but also the orange, blue, orange, green, and blue, they start to express the cytokeratin marker, which is for the differentiated population. So although the cells are differentiated, they are maintaining stemness because of their the gene expression pattern and signature, which I have just told you about. So our results so far suggested that there is red, which is a multipotent oral cancer stem cells. However, the cells are undergoing differentiation but still maintaining hybrid state of stemness in differentiating subpopulation. This was the result so far. We took help of Mohit to give us the unique differentially expressed gene, which is very unique to red, very unique to orange, very unique to green. They are not overexpressed, or these are not differentially expressed between the two populations, but among the four populations. And then they gave us this gene expression of 577 genes, which are uniquely expressed in red, and so on. So this unique gene expression signature was basically mapped against the gene expression pattern of oral cancer patients, which are there in the TCGA cohort, the cohort which is for the whole oral cavity cancer patients. And we found that if the patient has higher matching with the red signature, the gene signature which is uniquely expressed only in red, if they are having higher matching, they are surviving better. So this became counterintuitive. If on one hand, we are observing that the red is the most potent population and it has the property to generate all different kind of cells, then the patient which are showing the gene expression pattern similar to the red should have been showing the aggressive cancer, and they should have a poor prognosis. So we dig down and try to understood this phenomena, and what we are proposing here is that all those patients which will show a red signature will be those patients only where the red cells have not got the property to generate heterogeneous population. So in these patients, the red population is kind of blocked, epigenetically blocked. This is just the hypothesis which I am sharing with you. It is kind of blocked here in this state only, and they don't have in these patients, they are not producing all different kind of cells, and therefore they are matching with the red signature. Whereas if the patient has this kind of heterogeneity, then they will not match with the red signature, because here the cells, the tumor will be a mixed population and the red signature will be lost. So with the mixed population, there is poor prognosis, and therefore we are suggesting that the phenotypic heterogeneity translates to poor prognosis in gingivabuccal oral cancer. how do we know whether we can prove it or not? We have these cell lines in our hand where the GBC02, 029B, they are the more heterogeneous cell line where 24 negative and 24 positive cells are coexisting, whereas SE032, it is mostly 24 negative population, means it is just producing these population. So if you see there, the IC50 of 032 cells, they are in the range of 3 micromolar, whereas for the more heterogeneous cultures, the IC50 value is much higher. So this is just a preliminary thing to suggest you that the heterogeneity may be important, and so far which I could tell you that how a non-genetic heterogeneity can be achieved in a solid tumor. So as I had started telling that in a balanced ecosystem where the predator and prey are dependent on each other, our study has shown that the ecosystem which we are talking about or which we are trying to understand, which is in cancer, is not as simple as this. The energy does not flow between the predator and prey, whereas it can be as complex as this ecosystem where we can actually will not be able to identify our owl here sitting in a trunk because of their that can be because of the plasticity present in this species. So if it is present here, it will not be visible because it can actually mimic itself as a part of the system. So if we have to win our war against cancer, we need to identify these enemies which are disguised, and it is very hard to identify. It is not as easy to identify as we had thought earlier, that we will identify cancer stem cells as a phenotypic marker. Rather, now the cancer stem cells are coming out as more plastic. They can be existing in all different kind of shapes and colors. Therefore, probably now the new hallmark of cancer is including this phenotypic plasticity as one of the hallmarks, and that is suggesting, that may be suggesting that this is the way ahead for if we want to win war against cancer. So I'm just giving you the example of where we are leading from here is that we are showing that the notch low and notch high. Notch is also a gene which is very important for the proper development of the body, and the cells which are notch high by the expression of this transgene, or where the notch activity is blocked by the expression of this transgene, both are tumorigenic. And basically, if the cells have less the higher expression of notch low gene, then they are worse prognosis. So when we treated the cells with cisplatin, this is the culture of the 3D culture of cancer stem cells. When we treated these cultures with cisplatin, the notch expression has gone up in the cells. And then even when the cells were separated from here, they were allowed to grow again. They formed the secondary colonies, and the notch expression remained high. So basically, notch high state is maintaining the cisplatin tolerant state in the cancer. So if we block notch, so as a strategy, if we block notch by small molecule inhibitors of notch inhibitors, then we could actually sensitize these cells because by cisplatin. So basically, by blocking notch, we are providing the clue to the field that if notch was responsible for state transition for achieving cisplatin tolerance, we need to identify this kind of targets and block those pathways, and then give the treatment for blocking that plasticity, which is from red to the green. If we block that kind of plasticity by understanding the molecular targets, then we can sensitize our cells to commonly used chemotherapeutic molecules for better response. So the take-home message for today's talk can be that unexpectedly we found that oral stem-like cancer cells were very versatile and less compartmentalized. It cannot be identified by just one marker, which was basically proposed or thought previously. And cells acquire different functional state by conserved process of cell division. this pattern of cellular plasticity appeared to be unaffected by genomic differences in the cancer cells. So we are also providing the clue that this is the state change among the population cannot be as stochastic, because 24 positive negative cells are becoming 24 positive, but in our experience, we could not see any 24 positive cells are becoming 24 negative, which was actually proposed in a previous breast cancer paper, where any differentiated population is able to form any other colonies, any other markers. So this, if we understand the molecular switch responsible for plasticity in oral cancer, that may provide a novel therapeutic target against aggressive behavior of cancer. With that, I would like to say my acknowledgements to the collaborators from NIBMG, from Tata Medical Center, from ISA Kolkata, from NCCS, ISC, and the funding, generous funding provided by India Alliance, and intramodal funding by NIBMG. And this work is mainly the effort made by the Kavya. She is right now doing postdoc in Cleveland Clinic, USA, and the notch work was done by Subhashish, and this is my group members. So with that, I would like to thank again the organizer for giving me the time to present my data here today. So if there is any question, I can take now.
You are muted, Dr. Karthiki. Dr. Karthiki, you are muted.
Yeah, Karthiki, your mic is muted.
My apologies. Thank you, Dr. Sandeep, for that excellent talk. We learned a lot about stem cells and their role in cancer, and also the stem-like cells from your very elaborate presentation. Thank you very much for that. I will hand over to Dr. Alpana to see if there are any questions for you.
Sure. Thank you. Thank you.
So for all the attendees, you can type in your questions in the chat box, as I've mentioned already. In the YouTube chat box, I mean.
Maybe the attendees have to see the talk again and again on YouTube, and then they will put the questions in the comment box.
Yes, probably. So you can, if you, there is a question. So is there any difference of non-oncogenic cascades between the CD24 high, CD44 low, and ALDH high, and the CD24 low, CD44 high, ALDH high populations, leading to drug resistance?
Yeah, this is very specific question. So this phenotype which I am talking about, combining three markers, they are basically not that common in the literature. So people have worked on 44 positive ALDH high population and not combined with CD24. So right now I will not be able to tell you the oncogenic pathway which may be specifically upregulated or governing this specific phenotype. Our own data with Notch is suggesting that the ALDH high population, whether it is 24 positive or 24 negative, that is driven by the Notch activity. However, the cells become Notch low, Notch activity low when they are becoming ALDH low.
Any other questions? Well, if not, then thank you, Dr. Sandeep, for your time and for the excellent talk, and to all the attendees for joining us. If you have any questions after seeing the talk again, please feel free to mail us. Our email IDs are on our website, so we'll be happy to answer. Over to you, Dr. Karthiki. You're muted again, Dr. Karthik.
Sorry, I keep forgetting to unmute myself. I apologize. So thank you, Dr. Alpana. Thank you very much, Dr. Sandeep, for giving us a nice talk. As I mentioned before, I thank all the organizers, and I wish you all a pleasant evening. So goodbye for today.
Thank you. Thank you very much.